In this video, we will continue our discussion about young and aged CD8 plus T cells analysis. In the previous video, we have understood that in the case of this aged CD8 plus T cells, cells are more accumulated near neuron and blood-brain brain barriers. So they wanted to check what are the changes that occurs at this site compared to the young CD8 plus T cells. Since young CD8 plus T cells does not show any accumulations at this site, so they did the qPCR analysis of this 4 different genes that are associated with this blood-brain barriers and the neuron, and they are able to see that this synapsin 1, DLG4, CamkII, Homer1, all these are downregulated in the young. All these are downregulated in the aged CD8 plus T cells treated mice compared to the young. Afterwards, they also check the genes that is responsible for the response in the neuron, that is c-Fos. It is found to be high in the young CD8 plus T cells, whereas very low in the aged CD8 plus T cells. The quantitative analysis also confirm this immunohistochemistry ISC image, immunohistostaining. Still, I'm not able to recall this full form, but it's the ISC image. And when they also did the ISC imaging for other genes that are shown in the panel G and H, where this ACTA gene is found to be upregulated in the aged CD8 plus T cells. That can be seen here, that ACTA genes in the aged CD8 plus T cells are high in number, whereas CD31 and the collagen 1 are found to be lower in number. That can be seen in the panel H, that in the young CD8 plus T cells, this collagen 1 signals are high compared to the aged CD8 plus T cells. And also the CD31 signals are also high in the young CD8 plus T cells treated mice compared to the aged CD8 plus T cells. And this things is evident from the quantitative analysis that shows that the CD31 is significantly higher and the collagen 1 is also significantly higher. Afterwards, they checked this novel object recognition, which is a standard test to check the effect of this young and aged CD8 plus T cells in the young mice. So they are able to see that aged CD8 plus T cells has lower novel interactions, whereas the young CD8 plus T cells has high novel interactions. And afterwards, they check this learning and memory analysis, where they are able to, but this first 4 days belongs to the testing, where both of them performs almost at the same efficiencies, and this day one short-term memory testing is done, in which case we can see that this aged CD8 plus T cells has slightly more error. Afterwards, this day 2 testing is started from 6 to 10 days. In this case, we can see that error rates are slightly higher, for this aged CD8 plus T cells compared to the young CD8 plus T cells, as it follows a low error profile, whereas aged CD8 plus T cell treated mice follows a high error profile. So this in altogether, along with the methods that we have discussed in the previous video, shows that this aged CD8 plus T cells has a negative impact on the in terms of the response, brain response, and that could be related to the cognitive response. That, but by the end of this figure, they have not reached to the point of the cognition. They have just shown that this young CD8 plus T cells and the aged CD8 plus T cells has different impact when they are transduced into the mice, in a way that aged CD8 plus T cells shows decreased brain response, also have the decreased learning and decreased brain response through different results. One of them is this novel object recognitions, and other one is through this learning and memory test. And also these qPCR results also hint towards the same, that aged CD8 T cells has lower brain response or lower brain response here, lower brain uh, efficacies, not response, lower brain efficacies compared to the young CD8 T cells treated in the mice conditions. So that's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.